Hello everyone. Let's proceed on to chapter second of Basics of Anthropology. The name of chapter is Genetics and Evolution and the book is Ember and Ember. So Charles Darwin during the 19th century, he might have set off a new thought on genetics and evolution, but he was also unaware of a lot of things. So, for example, it seems clear that natural selection is not always a uniform process, but it can also act in jumps and starts. So, another example, scientists think that they have observed natural selection in action in British moths. So, in 1850, an almost black moth was spotted for the first time in Manchester, and it was quite unusual. For the most of the moths, they were grey, speckled grey. A century later, 95% of the moths in industrial parks of Britain were black. So only in the rural area, grey moths could be seen. So how to explain this phenomena? So the reason is, it seems that in the rural area, the grey speckled moth is hard to spot by bird predator against the lichen growing on the bark of trees. But in industrial area, lichen is killed by pollution. So the grey speckled moth, which were formerly well adapted to blend into their environment. So mm, this lichen and their grayish color, it act as camouflage. But because of industrialization and because of its subsequent manifestation such as killing of lichen, blackening of industries, pollution, the overall grayish and blackish environment, the camouflage cover it was blown away and they started becoming visible against the darker background. So it was easier to be to fall prey to birds or predators. In contrast, the black moths which previously had a disadvantage against the lighter bark, now they they become better adapted for survival, and this dark color of their wings it became an advantage, and subsequently the grayish population it became extinct or it became less in number, and the dark moths they became predominant. So what's the function here? What's the natural selection here? And scientists have found this as an example of natural selection. So let's observe two scenarios: pre-industrialized, industrialized, and industrialized. So these are type of environment, two type of environment that existed side by side or existed in chronological lineage. So in the pre-industrialized or unindustrialized scenario, the usual work of nature, it was that these were tree and on its bark they were lighter colored lichens, and because of that lighter background, the gray moths, they could easily camouflage themselves while sitting on it, while perching on it, and because of lighter background, the lichens, it act as camouflage. Camouflage, it means uh, they remain obscured, they remain hidden from the sight of predators. So here on pre-industrialized or unindustrialized society, natural selection, it favored light colored or grayish moths. And since in unindustrialized or pre-industrialized environment, lichens could easily grow, and because of their color, the because of the color which covered the bark of a tree, a moth could easily protect itself. A gray moth could, could easily survive itself, and when a particular individual can ensure its survival, it is likely to reproduce more. And in that scenario, its lineage, its species, it will survive. So ultimately, the whole species of gray color, they will likely to survive and reproduce more when there is no pollution, and when lichens are grayish in color, so will be the colors of gray moth, and that will provide camouflage. So this was the first instance of natural selection that was readily provided by nature itself. Then if we talk about industrialized countryside or industrialized area, now the the bark of trees of industrialized areas, first the lichens, if you do not know, then lichens are indicators of pollution. So if there is any area in which lichens are dying, it means the pollution level are high. So in the same way, so when the industrialization set in, lichens started dying. So this cover, it was blown. So lichen, if they started dying, the the trunk or the bark of tree, it will be exposed, which are always in darker in color. So now what happens, since the lighter color camouflage, it has blown away, and on any darker background, an entity or object of lighter color will become easily visible. So in the same sense, when the lichens got blown away, the gray moths, they started becoming visible when they spotted on any bark. So it also became easy for the predators to spot the gray moths and eat them. So this sudden change of atmosphere, which is a form of natural selection, it acted against the survival of gray moths. Now they could be preyed upon easily, and if a particular species is preyed upon that easy, so the predators, they will multiply in number. So it will be a vicious circle in which preys can be easily killed, and if preys can be easily killed, the predator, they have more food in their coffers, and if more food is available to them, so predator, they will multiply in number. And if there are more predators, so they are likely to eat more such visible prey. So here natural selection, it acted against the lighter color gray moths, and swiftly and gradually they started declining in number. Now another situation came. Uh, if you can recall the three principles of natural selection, first one out, which is variation, that any species and every individual, both species or population and individual, both of them, they possess variations in them, in their genetic code. Variations are always there, but sometimes some variations are not expressed. So some variations do not come out easily. the which in biological or genetical terminology it means expression. So if such traits, if they are not visible but they still stay in that in their codes, in their lineage, and we will talk about more in further chapters when we are going to talk about dominant and recessive traits. So let me give an idea. Dominant traits are those traits in a particular species or an individual, those traits that get expressed easily, or the traits which out of two out of two different alleles or two different traits, they get expressed easily. So those are called dominant, and those dominant traits, they are likely to be visible. They are likely to dominate the expression, whatever the genetic composition of a particular species or individual is. So let's say uh, a, a particular individual, he possesses two variants of tallness. One is dominant, which is taller, and the other one is shorter one, that is recessive one. So now the taller one, this is a dominant trait. So if a particular individual has this trait in the genetic code, so this trait is likely to be expressed, and the other one, it is um, likely to hidden away or likely to remain hidden. But if a population contains or any individual possesses both the traits which are recessive in nature, then this recessive, it is going to come out, it is going to be expressed. Otherwise, all such heterogenomes or all such hetero individuals in which the composition is ordered in this way, in which one trait is dominant, at least one trait is dominant, then this dominant trait is going to express. So my point is, variations are there, they remain hidden, and I have talked about one particular trait, and let me clarify each particular trait in the whole genomic code or the whole genetic code. So each trait, it is regulated by a set of genes, and it is also acted upon by environment. So it's a complex phenomena. I might have made this simple in terms of one particular trait, and this expression, it is much more complex than what I have described here. But yes, the process, if rooted out and if simplified, it will be expressed or it will be explained in this way only. So coming back to the point, variations are there. So in the same way, in the British area, whether it was industrialized, pre-industrialized or unindustrialized, in all the areas, there were variations. So there were gray moths, there were black moths, there were moths with uh, dark shades of gray, there were moths with different colors, lighter shades of other colors. So such variations were there. But since nature decides which trait will go, so in these two society, these are not society, um, sorry, I mean these are environment. If we look them through the angle of biology and genetics, so these are not just society, they are actually environment. So unindustrialized society is an environment in itself, the same way industrialized society is. So here the environment or the nature, it will act on the particular variants and the expression of a particular variant, it depends on the environment a particular species in. So in unindustrialized, dark dark color or any variant of dark color which cannot camouflage itself, they are likely to be easily preyed upon. So the unindustrialized society, it favors gray moths. So they cannot be spotted among the lighter background of lichens, and this makes them hidden. This makes their survival possible, and since their survival is possible, they will multiply in number, and this is how this particular species will remain dominant in a particular environment. But other variations will be there, though they will be um, way less in number than the dominant one. And in the same way, in industrialized society, now the camouflage, it has been removed, and light color, the grayish moth, they can be easily preyed upon, and when they are preyed, the predators, they will increase in number, which will negatively affect them. And in such a scenario, the natural selection, it comes into picture, that variations are there. And now same different variants of moths, they are sitting on bark. The gray will be easily spotted and killed or eaten away. Other lighter colors will also be eaten away. It will be on their disadvantage that they have such color. But the dark gray or the darker colors, the black or dark gray, both these variants, they will not be spotted against the change in atmosphere. Now, against the dark colored bark, dark bark in industrialized countries, especially the industrialized societies of 18th and 19th century, because of pollution, almost all the tree species, all the buildings, they were covered with soot and carbon, which gave them dissipatingly dark or grayish color, dark gray color. So in such a scenario, the dark moths, they will not be preyed upon. If they are not preyed upon or they cannot be spotted easily, it will multiply their number, and their survival, it will be ensured. And moreover, the predators, they are anyway going to eat away the grayish one, the lighter color ones. So they are not going to compete for resources against any such a species, and they will become the dominant one. This is how natural selection acts, and this is how the example, this particular example, British moth, have been spotted. So this dark color was earlier it was a disadvantage, but now with the change of environment, it became an advantage, and subsequently darker moths, they became predominant in that particular atmosphere. And this transformation, it did not just occur in Britain, it also happened in other highly polluted areas such as Ruhr area of Germany or Pittsburgh of United States. And in the same way, all such expression of working example of natural selection have been observed. So the type of natural selection in moth species, it is known as directional selection. So let's see the three types of natural selection. So directional selection is a selection that increases the frequency of trait that is positively favored. So in unindustrialized area, it is the gray or lighter color, and in industrialized area, it is the darker variant that has been favored. Now let's talk about normalizing selection. So this type of selection, it removes harmful genes that arose by mutations. And then the balancing selection. It is when a heterozygous combination of allele is positively favored even though homozygous combination is disfavored. So this type of selection, balancing selection, it means it it favors average or intermediate individual. So this natural selection which has been spotted in moth is direction selection because a particular trait has been positively favored. But natural selection can also take other forms such as normalizing selection and balancing selection. So in this, the average value does not change, but natural selection, it removes the extremes. So an example of which is birth weight of babies. So the extremes such as both very low birth and very high birth are disadvantageous and they will not be selected against since very low birth it this expression it is going to produce children which are malnourished and such individuals are not likely to survive. And if birth weight is very high, it is also be very difficult to be carried in uterus for long, and it will also be difficult in the on many counts such as nutrition. Then it will also be difficult while giving birth. So the extremes, they are not selected against and they are removed in normalizing selection. Then comes the third possibility, that is balancing selection. So it occurs when a heterozygous combination of alleles is positively favored even though homozygous are disfavored. So this particular is example of this one. So if a particular species, so here heterozygous, it means two different variants of alleles are there in a particular individual. Now if both the variants are same, they will be known as homozygous. So this homozygous variant in which both similar traits are there, they will be disfavored, and heterozygous, they will be favored. This is known as balancing selection. Another example of observed natural selection is acquired resistance of housefly to insecticide DDT. So DDT is a natural chemical pesticide. So when its use started in 1940s, several new DDT resistant strains of housefly they evolved. And even though the early DDT use it killed many such houseflies, but a few survived, and those that survived were those that build resistance to the application of DDT. And this also has become a newer threat in the present time when microorganisms, especially the bacteria, they have built resistance against the use of antibiotics. So a particular antibiotic may lose its effectiveness after it comes into wider use because new strain of bacteria will emerge and the earlier antibiotic and its chemical composition, it will not work on the newer strains. So let's have a look at both the scenario. First is DDT resistance. So DDT is a pesticide, a housefly killer. And now let's see how natural selection work on it. So let's say there is a population of these many houseflies in a particular area, and as per the mechanism of evolution, mechanism of life, the genetic code, so it seems that these many individuals, they are not homozygous, or they are not similar in their genetic composition. They are not similar, dissimilar, and because of this property, variations are there. So in this pool of housefly, different kinds of different variants, different traits, different allele combination. So all such will exist in this pool of population. And depending on the environment, some traits will be observed, some traits will become dominant, they will be expressed, and while the rest, which are not seen easily, they will remain hidden in these population. So here we can see all the individual in this particular piece pool of housefly, they do not have similar genetic composition. Variations are there, even if they are not visible, even if they are not expressed. So at first, when DDT was applied to this pool of housefly, so let's say it killed most of the housefly, but a few strains which were not expressed, but such variations were there. So these strains remained. Now this particular DDT, with its chemical composition and the chemical that are in it, those chemicals, they might have killed 90% of population, 90% of the pool, but still a few 10% variations, 10% of the species of the pool, they survive. Now this the remaining one, they are likely to reproduce, and since variations were there which could not be eliminated the variation, it means elimination of the genetic code itself, which is not possible, or alteration of genetic code. So the application of DDT, now the remaining individual, the they will not be threatened by, they will not be acted upon by any further use of DDT, since these species, they have built resistance against the use of this particular type of DDT. So this the more you use it, it is not going to have any adverse effect on this pool of population. And since housefly and any such species that comes in micro or small scale, they have a very they have small lifespan, which means these individuals, they will reproduce in faster way. So maybe in a few days they may produce 10 or 20 generations. Let's compare it with the lifespan of human. So a human in present time, so we say we started reproducing around an average age of 25. So those first 25 years, they are artificially infertile, because even if the onset of puberty, it sets around 14-15 years, still we wait for 10 years. So those 10 years are infertile years. Then The first offspring, it is given expression on 25th year. Then we can say by 30 we have 3 kids. So let's assume this to be an average. So by 30 you have 3 kids, or put it in another way. Suppose someone is born in the year 2000. His first offspring will be in 2025, and the same offspring, he will become he will have another offspring in 2050, and his offspring, they will be able to procreate in 2075. So if we take the overall example in number of generations, so here they are uh, the gap of 25 years for a generation. This is the reproductive span of human species. 25 years for a generation. And for true generation, we might have 50 years. I'm not talking about multiplication of individual, but generation. So in 50 years, we have crossed only 2 generations. But the the species which are on the micro scale or on the small scale, they have very shorter reproductive span. So they may be able to produce 10 to 20 generation in a few days. So by this, the pool of the remaining survivors of DDT exposure, they are going to reproduce, and in a few days this pool, it will be filled up again. And now let's say in a month they have reproduced and come up with 10 or 20 generations, and now this pool, it has been refilled again, but with DDT resistant housefly. Now this pool has those houseflies which cannot be killed by the particular strain of DDT. So here natural selection favored those variants, those survivors which can ensure the survival of their species, survival of their life form, even after application of a particular strain of DDT. So this is how natural selection was observed in one another example. And now this same example, it has been circling around in news in the form of antimicrobial resistance, which can become a newer pandemic, a newer global scale threat. So same variation, same principle, same phenomena works here. Let's say this is a particular pool of bacteria. Now any antibiotic is given or employed to remove all these individuals. So say X is an antibiotic which has been used to act upon each of them. So this antibiotic, it will kill even the 99% population, but a few will remain. Let's say if 1% remain, and 1% bacteria are those that have a survival trait, a variant that help them survive, help them build immunity against the application of antibiotics. Now again the same process will happen. These are microorganisms, so their reproductive span will be way less than the housefly. Let's assume it is that in a few hours these bacteria, they are going to have 10 to 20 generations. So those survivors, they will be selected by nature to reproduce. All the other variants which could not survive the onslaught of antibiotic, they will be wiped away. And but the survivor which has built resistance to this particular class of antibiotic, they will survive, they will reproduce, and at this scale, the reproduction is very rapid, very fast, and in just a few hours or a few days, they will refill the pool just as the housefly did. And the same problem is occurring in the antimicrobial. So here, the problem is the development of antibiotics, it takes a lot of time, lot of research, lot of trials, lot of money and funding, and the development of resistant bacteria, it is a work of a few days. So the issue is with the time lag. Since newer strain of antibiotics, they take lot of time to develop, but newer strain of resistant or antibiotic resistant bacteria, they can emerge in a few days. So what happens is, when a particular group of bacteria, if they build resistance against a particular class of bacteria, so let's say earlier they build resistance to the X strain, now the pool has been refilled. So this is the pool of the bacteria that have survived the killing action of antibiotic X. So now we have a higher antibiotic Y, which is higher in potency. Now it has been applied to this pool of bacteria, and the same process will happen, that 99% population will be eliminated, but 1% strain will remain. So this 1% pool, it will build resistance and will reproduce and refill the pool in a few days. So again, we have come up with a higher strain of antibiotic use, and now again the same process continues. So bacteria, they are building resistance to all possible classes of antibiotic use, and this is a cause of concern, since the development of antibiotics, it takes time, but resistance in bacteria, it is happening, the reproductive span is less, so the strain of bacteria that build resistant to it, they reproduce and refill the pool in a few days. So there is a whole time gap, which is a cause of concern, that what might happen if we run out of a new strain of bacteria, a newer strain of antibiotics, but the bacteria, they start proliferating in exponential way. Then let's suppose we come up against a situation in which the highest class of bacteria, sorry, not bacteria, antibiotics, so a higher class of antibiotics such as third or fourth generation. So you might have got this idea here. When I talk about third and fourth generation of antibiotic, it means the first and second generation of antibiotics, they are proving less effective. They have lost the edge. So effectiveness is is gradually losing its ground. And since bacteria, they are developing resistance, so it's not further from the truth that these strain of bacteria, third and fourth generation, and even the most potent one, carbapenem, one day, if this resistance, if it is not stopped, so the highest class of antibiotic may lose their effectiveness, and that will be a scary situation. Now, since a particular antibiotic may lose its effectiveness after it come into wide use because new and resistant bacterial strain emerge. And one possible way to deal with the problems to stop and using antibiotics for a few years. Yes, this can happen. If you stop antibiotics for a few years, then the according to those lower in reproductive span of antimicrobial, sorry, lower reproductive span of bacteria, the the dominant or resistant variant, it is going to lose its edge. It will not be selected if for a few years the application of antibiotics is not there. So this application of antibiotic, it is the natural condition that is selecting the resistant variant. But if the application itself is stopped, the resistant variant, it will not be selected against. Then the appearance of a beneficial trait may assist the survival of an organism. But what happens when the organism reproduce by mating with members that do not possess this variation? So will the new adaptive traits eventually disappear if subsequent generation mate with individuals that lack this trait? So the shorter example is given here. So if an organism that has that possess a dominant or say the beneficial trait which is T, and this individual is mated with the other individual which do not possess that trait, and the resultant child will have this heterozygous combination. But even if the the, the offspring or the subsequent generation, they are holding that trait which is not beneficial, genetics or inheritance or natural selection, it says there that if any individual is heterozygous and a dominant variation is there, this dominant variant, it will be expressed. So even if the dominant they mate with the recessive and the offspring it possess both the variants, only the dominant it will be expressed. Let's see how it works. Now let's proceed on to heredity. You might have heard about Gregor Mendel. He was a monk, an amateur botanist who worked with varieties of pea plants, and he chose the breeding partners that differ by observable trait. So by his experiment, he came to give the idea of genetics, of evolution, of hereditary. So he chose the plant that differed in observable traits. So observable traits are those traits which can be distinguished, which distinguished by their appearance. So, such as he bred tall plants with the short one and yellow with the green one. So when the pollen from a yellow plant it was transferred to a green plant, Mendel observed that all of the first generation they bore yellow peas. It seemed that the green trait had disappeared, even though he believed that some of them they are going to be greener. But when the offspring they mated with the first generation, so here the heterozygous which possess both the variants of yellow and green, let's say at first he bred the plants in this way. So the offspring they possess the variants in this way. So these are homozygous. This is homozygous dominant. This is homozygous. recessive. And these first generation all were heterozygous, in which yellow was dominant and green was recessive. Dominant is written in capital letter, recessive it is in small letter. So this alteration of letter itself shows that the capital one is the dominant one. So the first generation, even they even if they had the recessive traits in them, only the dominant one was expressed. So all the flowers, they were yellowish in color, even if the green color was there. But when he bred this one with the first generation, they produced both yellow and green in the ratio of 3 yellow and 1 green plant. Let's look at it here. So this was these were they were dominant, but these this was dominant homozygous and this was recessive homozygous. Homozygous means they have both similar alleles. Homo and hetero. So homozygous alleles are those that have similar traits such as YY, TT, RR, red, and heterozygous which possess one dominant and one recessive. So here recessives can also be homozygous. But if one is dominant and the other one it is recessive, it is known as heterozygous. So this one also is a heterozygous. So both of them they are homozygous. This is homozygous dominant and this one is homozygous recessive. And when they were crossed amongst one another, the subsequent generation it has the composition in which the dominant variant it appeared in all of them. And now when this first generation, when it was crossed between itself, and let me clarify one more thing, that even though the yellow plant it was it was mated with green plant, or the yellow color was mated with crossed with green color, the expression, all of them, in all the subsequent generation, it was yellow. So it showed that yellow is the dominant color, and irrespective of the other allele, if dominant is there, it is going to be expressed. And the same way recessive one, if recessive is there, it will be expressed only in the homo situation, in the homozygous situation in which both the variants are green, then only it is expressed as green. Otherwise, if one trait of dominant is there, the dominant will be expressed. So both these plants they are yellow. Now both the heterozygous they were crossed among themselves, and now the newer variation will be in this way. So you can cross it in this way. First Y and Y variant will give homozygous dominant, then Y with the recessive green will give heterozygous, then G with Y it will also be a heterozygous, then G with G will give a homozygous. So when crossed among themselves, the next generation, F2 generation, it will come out or will be expressed in this way, in which this one it will be yellow, this one it will be yellow, and this one also it will be yellow. As I have told you that yellow is a dominant color, and in either in a homozygous dominant or heterozygous dominant, in both the condition if at least one dominant trait is there, it will be expressed. So in all the three offspring, first, second and third, the dominant variant is there, so it is going to be expressed, and the color of all first, second and third offspring, it will be yellow. But recessive genes such as green is recessive, and it will be expressed only in a homozygous condition, which means if both the variants are homozygous or both the variants are recessive, then only it will be expressed, and the color of it, the fourth offspring, will be green. So this ratio 3:1, it was identified by Mendel, 3:1 ratio. Now, from his result, Mendel concluded that some yellow plants were pure homozygous, and others also possess a green factor, which was heterozygous. So, homozygous dominant, this one, and heterozygous dominant will be this one. And although two plants might both have yellow peas, one of them might produce offspring with green peas. In such genetic makeup, the genotype differed from observable appearance. So this is the confusion behind the variation, which is the first principle of natural selection. That variations are there because at a time, the phenotype is how a particular species is expressed or how a trait is expressed. So expression of a trait does not clearly portray the makeup of genotype. So here in this case as well, so these three offspring, first, second, and third, they all differed in their genotypes. Genotypes is the genetic makeup, is the genetic code or the variants, traits, genetic makeup or genetic code of traits. So if you see first, second and third offspring, the first one has both the dominant traits. It is the genetic makeup of the first offspring. And if you see the second one, now the genetic makeup of both first and second are different. So their genotype is different. Yet both of them, they are yellow in color. So yellow, it is the color which is expressed, which can be which is visible, which can be observed. So this observable trait or the appearance, the visible trait, it is known as phenotype. So this is where the variations upon which natural selection act, they lie. So here you can see, even if you have different genotype, you can have similar phenotype. So this was the observation that was made by Mendel. So here, first generation in which YY, both the capital, both the dominant one, but since both the traits are similar, it is known as homozygous. And here, the first generation by which the dominant homozygous was crossed with, it was also a recessive homozygous because it possessed both the similar trait. When they were crossed, the first generation, Y with Y, this one, and this Y with this Y. So the first generation, they were heterozygous. The genotype, it changed. The parental generation, they were homozygous. They were pure traits, both of them. But the offspring, their genotype, it is heterogeneous or heterozygous, in which both of them possess one dominant and one recessive trait. And when this generation it was crossed unto itself, the second generation, the shuffling of genotype, it happened in this way, that this one when crossed with this produced a homozygous dominant. This one when crossed with this produced a heterozygous, and again this and this with heterozygous dominant, and then the last one, it is homozygous recessive. So in the second generation itself, you can see the in the first three offspring, the genotype is different. This one is dominant homozygous, and both of them, they are dominant heterozygous, but yet the expression or the phenotype, it is similar. So this was what found out by Gregor Mendel. Now let's proceed ahead. Genes, the conveyor of inherited traits. So Mendel's unit of heredity were now known or called as genes, and he concluded that these units occurred in pair for each trait, and offspring inherited one unit of pair from each parent. So this was his finding, that these traits, they do not um, do not occur individually but in pairs, because each parent generation was also an offspring of their parents. So, such as the parents of this generation is this one. Say similar, the parents of this generation is the first generation. So the parent generation itself in this diagram, it was also an offspring sometime, and this was how that they come to derive the traits in pairs. So each member of a gene pair, it is called an allele, and if two genes or alleles are same, it is known as homozygous, and if they differ, it is known as heterozygous. So this one, the pair here, which has been derived from their parents, one from father, one from mother. So if this composition is similar, it is homozygous. If it is different, it is heterozygous. And a pea plant that contains a pair of genes for yellow, it is homozygous for this trait, and a yellow pea plant with dominant gene for yellow and recessive, it is heterozygous. So the genes of higher organisms, which does not include bacteria and primitive plants such as blue-green algae. So our genes, it is located on the chromosomes. Chromosomes are located in the nucleus, and nucleus are in cells. So cell, which is the primary unit, it possesses both nucleus, DNA, and then chromosomes. And the traits or the genes which we are talking about here, in higher organisms, including humans, they are located on chromosomes. So chromosomes usually occur in pairs. Same way, you can, if you want to draw a diagram, most of the higher organisms will be drawn in this way. Since we, most of the higher organisms, they are derived, their genetic composition, it is derived from both of their parents, both from father and mother. So in that sense, they are derived one copy from each of them. So that's why our genes are in you can say in couple, in dual, in twos, or in pair, and each allele for a given trait is carried in the identical position on the corresponding. So this will be seen in further chapter on genetic and evolution. Let's see what mitosis and meiosis are. Let me clarify one thing. Chromosomes, genes, their structures, their pairing. This this is a long chapter, and it needs further clarification. And right now, I believe when I'm going through basics, it is not necessary to go through the description of all the elements, since I can explain basic things such as what genotype, phenotype, genes, and homozygous, heterozygous, um, such concepts can be explained because they are really simple in task. But to explain um, the chromosomal dexterity, the chromosomal situation, how do they align, what chromatin is, and how do they pair. So it needs a bit of time. So in the basics, let's keep ourselves to have an overview of what genetics and evolution is. But yes, when time comes and when the chapter comes, we are going to talk about them in real details. And let's proceed to mitosis and meiosis. So body cells of every plant or animal, they carry chromosomal pairs. And the pairing mechanism here, I have explained this. You might have get this clear idea that all the plants and animals, we are product of crossovers. So crossover is between father and mothers or a parent organization, which themselves are two in number. So we are going to we are going to derive one copy from each of our parent. So that's why genetic composition or the traits, the basic traits, they are always in pairs, 1 and 2, derived from each of the parent. So human have 23 pairs, so with total 46 chromosomes, and each carrying many times that number of genes. And each new body cell, it receives this number of chromosomes during cellular reproduction or mitosis. So most of these cells, other than the gametes, they divide through a process called mitosis. So here, mitosis in plant cell is given here in diagram. This image shows the metaphase. This is a stage of cell division. So the chromosome that are stained dark red here, these are the chromosomes that are bundled up here. And while the division, when it proceeds to its final stage, these chromosomes, they get separated and align themselves on the edges. And then when they are clearly aligned to the edges or the ends, this particular cell, it is going to divide from half, and then we will have two new cells. This is how mitosis work. So this is another example of cell division, but this is in this process, meiosis, it happens in gametes, while mitosis, it is a process of all other cells which are not gametes. Then what happens when a sperm cell or egg cell unite to form a new organism? So what prevents the human baby from receiving twice the number of chromosomes, 23 from sperm and 23 from egg, which means why can't we receive 46 pairs, 23 from father and 23 from mother? So the process by which reproductive cells or gametes are formed, it is known as meiosis. And this meiosis, it happens that division will or should occur in this way, not just should, it must occur in this way, that every individual derive only 23 pairs out of 46 pairs that are available. So this division, this halfway measure, it is ensured by this process meiosis. So each reproductive cell, it contains half the number of chromosomes appropriate for this species, and one member of each chromosome pair is carried in every egg or sperm. So nature itself, so in all other cells, bodily cell, there are 23 pairs that a particular individual has derived from his parents or its parents. But in the gametes, in the sex cells, which are sperm and egg, only one copy of these chromosomes it exists. So, so nature itself, it distinguishes between bodily cells and gametes. So that's why the composition, the chromosomal composition of eggs and sperm both are different. So sperm, the egg, it is XX, and sperm, it is XY. At fertilization, the human embryo, it normally receives 23 pairs from its mother and same number of its father, which adds to 23 pairs. So in in the egg, which is known as X, and in sperm, which is known as Y. So when they are inherited, this is how an offspring is going to be distinguished as a son or a daughter, a girl or a boy. And DNA, they are located on chromosomes. So each gene, it carries a set of instructional coded in it's a chemical structure, and this coded information carried in that gene that makes the rest of structural part and chemical machinery. Let me elaborate it. The chromosomes, they are structural part. So in sense, they by themselves, they do not possess anything. They are just some structures. And on those structures, when they are joined together and when they are pulled together, so this pulling together, it leads to the expression of characteristics. In simply, DNA, it is nothing but AT, CG, CGAT, AT, TA, CG, GC. So this whole code, if you look at it, it is nothing but the expression of these four bases, A, T, and CG, and they will be joined together in forward or obverse motion. So it may be AT and TA, then it's CG and GC, and this chain will go on. So this is just the structural part, same as the chromosome, which are nothing but structure. But the pooling of these structures or pooling of these codes, they can come to have a particular gene or a particular characteristics, which is called a gene in biology. So let's say this one is first position, this one is second, the 4, 5, 6, 7. Now these structural constituents which are abiotic in nature, abiotic in philosophical sense. It means by themselves AT means nothing. But when pulled together, let's say 1 to 4. So this part, this position, when pulled together, it may have to express something. For example, this particular chain, it is going to express the color of eye. Then let's say from 8 to 15, it might have, it may come to express height or length or extremities. So concept here is that the chromosomes, while we have talked about both mitosis and meiosis, so there may be some confusion regarding what those two processes are and what other cells and sex cells or gametes are. The difference lies in the inheritance, and the real difference, it is a complex of two phenomena here. So these two phenomena, they can be divided into, or the complex understanding, it can be divided into two parts. So our whole body, it can be divided into sex cells and the other cells. So Here the difference is that the basic unit of any organism is cell, a particular cell. When cell unite, they form tissue, and many tissue they unite to form organ, and many organ they form unite to form an organism, and many organisms they unite to form a population, many such population that unite to form a species. So all in all, the basic unit is cell, and this cell, it contains nucleus plus DNA, the whole machinery. And this DNA, it is in the form of chromosomes, and chromosomes are nothing but this structure. Let's say uh, this one structure, by which I mean that every offspring, it derives its genetic composition from its parents. So that's why the genetic composition of every such individual, it is in pairs, and the chromosomes are nothing but this pairing, but yet the structure it differs. So the structure it might be different in simple organisms such as bacteria. In microorganisms, they may not have such complex chromosomal structure, but yes, the pairing is there, and the chromosome they will be there. So these chromosomes, they exist in pairs, and this pairing of chromosome itself, it will mean nothing unless such chromosomes they give rise to a trait. So we all we look for is a trait or a characteristic, a feature. So herein, let's say that we have derived 23 pairs of chromosomes, and these 23 they can be divided into 22 pairs plus gametes. So they come together to form the 23 pairs which every individual, every higher organism they possess. Now what I mean is that these chromosomes, they are nothing but this structure. So DNA, which is chromosomes are nothing but condensed form of DNA, and DNA is the code that is formed out of repetition of base units. So these AT, TA, CG, these are the base units, and these when repeated in any such form, they form DNA, and DNA when condensed, they give rise to chromosomes. So this condensed structure, which is a mechanism of nature or the work of nature to preserve, to keep the DNA intact in its form. And such DNA, such repetition of basic units, it exists in every cell, in every nucleus. Now let's see here other cells and sex cells. So as I have said, every individual is 22 pairs of other cells plus gametes, which can be X or Y, which distinguish itself from the other cells. Now mitosis is about division of other cells, is division of 22 pairs of cells, which is necessary for many purposes. 22 pairs of other cells, and this division is necessary. Cells keep dividing even if when I speak finish, my body will have killed zillions of cells, and zillions of newer cells would have developed, and they would have killed and developed. So this process, it keeps going on, this division, it keeps going on, and this division is necessary. But meiosis, this is different, since first the formation of new organism, because that is the ultimate basis of all such natural phenomena, that a new organism should appear. And if we divide an organism by this method, by mitosis, which is very simple, one cell, it is divided into two cells, and two into many cells, and new cells keep dividing, older cells, they keep getting destroyed, and this is how the physical process, it keeps going on. But this is very simple mechanism, and by which, if the same process happens in meiosis or in gamete cells, it will create um, issues, biological and fertility related issues, which we will talk about in further chapters, in later chapters. But right now, all you need to know is that for meiosis, or for a new organism to form, the process itself, it need to remain intact. And then the the gamete cell or the sex cell, they also need to inherit a particular trait from their parents, since these two parents, each of them, they have 23 pairs. And now, if an offspring is getting all the pairs, the sex cell, they're likely to derive 46 pairs, which will make it unsustainable in the long run. Since if they are 46 pair, the upcoming newer mate, it will have 46, and this process, it will go up to, it may be very different, 92 pairs. So this process itself becomes futile and unsustainable. And to simplify it and keep the formation of organism, the process intact, meiosis process or meiosis type of cell division, it comes to our rescue. Now what happens, you have 23 pairs, the parents have 23 pairs, and now when fertilized, the organism, he is deriving only one copy, he or she is deriving only one copy from each of their parent. So this is how the 23 pairs are ensured. And now which copy is being derived, this is a matter of assortment, and this is a matter of natural selection, which we are going to talk about in this particular chapter. So when a sperm cell or an egg, they unite to form a new organism, so what prevent a human baby from receiving twice the number of chromosomes, that is 46 pair, is the process known as meiosis. So each reproductive cell, it contains the half of number of chromosomes that appropriate for such species. So one member of each chromosome pair is carried in every egg or sperm. So that's how humans receive 23 pairs. And let's see it further. Let's see the process of meiosis. So this is the condensed form of chromosomes known as chromatin. So at first, nuclear DNA forms into chromosomes and replicates, so which gives homologous pairs. This Such homologous pairs are formed, and now these pairs, they line up. So, and by lining here, crossing over may occur. Crossing over is kind of assortment in which the desired trait, it is taken up, or the trait which is missing, which may be available in the other unit, they can also be taken up. Then this segregation occurs in the diagram 4, and by this segregation, so equal number of and equal characteristic of these alleles or these chromosomes, they get lined up, they segregate, and this unique assortment is derived from each of their parents. And now these cells, they divide in the fifth one. Then in the sixth diagram, the daughter cells, both of them, they separate, and while separating each one of them, they have different assortment of chromosomes, which you can see here. This is how meiosis happens. Now you can see here the actual representation of DNA. So this is one such chromosome, which is the condensed form of DNA. If you unfold it further, you will see this structure. If you straighten this double helix, you will see a ladder-like structure in which bases are arranged in a repetitive unit. And if you further unfold it, you will see how bases are organized here. So both the sides of this particular ladder, they are phosphate and sugar groups, which held these bases um, in their straightened form. Let me explain it in simple way. This is the diagram B. This one is the double helical structure of DNA, and when you straighten it, it becomes a ladder-like structure. So here, any ladder, it is supported by these handles and these pedestals. Handles are those which help you to hold them and move up, and pedestal is the podium upon which you set your foot up and move upstairs. So in this ladder-like structure, these sides, these handles, these are the phosphate and sugar groups, which both of them on both sides, they are there to stabilize the middle section, the ladder one. And the pedestals, they are the bases AT, CG, CG, TA. So this is how DNA is organized. Chromosomes are built of DNA, which consist of two spiral sugar phosphate strands. These handles, these strands are sugar phosphate P and S, and linked by a nitrogenous base. These bases are nitrogenous, which are adenine, guanine, thymine, and cytosine. When a DNA molecule reproduces, the bases, they separate. So during reproduction, these bases, the double bonds, they get separate, and the DNA, it is unwound. And because adenine can only bind to thymine and cytosine only bind to guanine, each original strand serves as a mold upon which new kind of complementary chain is formed. And we will discuss about it further in later chapters, how complementary chains are formed. But right now, for you is the unfolded structure of chromosomes is represented here, and these bases, they are severed here when reproductive cells or meiosis happens, and when severed, A will never bind with G, or in other words, complementary of A is T. So adenine will bind only with thymine. In the same way, C will only bind with G. So these complementary pairs, they will come to help when these say these severed or these unbounded strand, if they are formed again, so these complementary pairs, they will attach again and the whole chain will be formed. Then genetic information, it is stored in the linear sequence of the bases, which is linear sequence is this A, T, T, A, C, G, G, C. This is the linear sequence of these pedestal, these bases, which is, if we straighten up, forms a linear chain. And on these bases, genetic information is stored. And this complementary pairs, here they are explained that DNA molecule, they always have the same opposite base. Adenine and thymine are pairs C and G, and that two strain that carry the same information. So when the double helix unwind, each strain can form a template for new strain of complementary bases. So here the information, it is similar, same information. So when it is severed, it is uh, I'm I'm trying to explain it with a familiar phenomena. So if this particular unwound section of double helix, if it is cut in the middle, then we will have both the severed structure, this on the left side, this one on the right side. Now the law of genetic evolution, it says that all these pairs, they are nothing, they are repetitive units and they carry the same information. So even on their even if they are cut into two parts, the information stored, it will be same. And then the other principle is that A will only bind with T. So in such a case, even if it is severed, the template upon which new pairs can be added, it will not be lost. So at any time if you want to join it, so all you have to do is to bring A with T. And since it is not that the information on the left side was something different than the right side, both of them possess the similar information, so they can be unwound and they can be wound again anytime. So this is the beauty of the double helix. Then Human Genome Project, it was started to map the genetic makeup of human. Then we are going to talk about messenger RNA in the further chapters. That's all for now.